Hello everyone, welcome back to my channel. Today we are going to discuss about histology of placenta and umbilical cord. Placenta is a disc-shaped feto-maternal organ. So it has a fetal side and the maternal side. So first we'll discuss about the fetal side. So fetal side is smooth, covered by amniotic membrane. So amniotic membrane we are going to represent in the form of a epithelium, which is usually the low cuboidal. Some books says it is squamous epithelium also. So that's the amniotic membrane. Here I am showing with a low cuboidal epithelium with a round nucleus. So deep to the amniotic membrane we have a connective tissue. That's the extra embryonic mesoderm. The mesoderm is represented by the loose connective tissues there, and this is cytotrophoblastic layer. The cytotrophoblastic layer which extend into the maternal side as along with the stem villi. So cytotrophoblast also consists of cuboidal epithelium. Even the extra embryonic mesoderm also extend along the stem villi, and through the branches of the villi they will be present within the center of the tertiary villi. So cytotrophoblast, some places they extend from the fetal side until the maternal side. That's the basic one which extend from the one end to the other. It is called the stem villi. So in one of the diagram, one stem villi we can show. So cytotrophoblast is a layer of cuboidal epithelium. As we are showing mature placenta, we are showing the tertiary villi, so which consists of a central mesoderm and lined by cytotrophablastic layer. So one stem villi I am showing here. The mesoderm also contain fetal blood capillaries. You can't differentiate arteries and veins, only the capillaries, the small capillaries which are distinguished in the mesoderm. These are the fetal capillaries. Outside the cytotrophoblast, we have syncytiotrophoblast. These are also cuboidal epithelium, but the cell borders are very indistinct. You can't make out the cell borders. So it just has a sheet of cytoplasm with a round cuboidal nuclei. That is a syncytiotrophoblast. So the amniotic membrane, the extra embryonic mesoderm containing the fetal capillaries, the cytotrophoblastic cell and syncytiotrophablastic cells constitute the fetal end of the placenta. Coming to the maternal end, also called the basal plate. So we have a cytotrophoblastic sheet. It is formed the same cytotrophoblast. Once it reaches the maternal side, it spread out laterally and form a sheet-like structure. So that is the same cuboidal epithelium we are going to draw. On this side we have a maternal tissue, the decidual cells. So that is, decidual cells are spindle-shaped cells. They'll be present scattered or in clusters. They'll have epithelial appearance. They also have a maternal blood vessels, and we are a septa. The fibrous septa extend into the fetal side. This fibrous septa is the one which constitute the cotyledons in the mature placenta, what we see. And because of it, there is no amniotic or the smooth membrane there. The maternal side is very rough appearance in the when we see the placenta. So this is cytotrophoblastic sheath, the same cuboidal epithelium, and the maternal side decidual cells and the maternal blood vessels. In between the fetal and the maternal side, we have the intervillous space, the villi of varying sizes. The branching of the villi we'll see. So these are the villi. So first we shade the background which represent the maternal blood will be there in the intervillous space. So these are the basal lamina of the trophoblast, means the villi. There will be varying different sizes. It can be a small, because the villi will branch, it can be of different sizes. So we have a cytotrophoblastic lamina, that is a basal lamina. Over that I am showing the syncytiotrophoblasts. There is no cell membrane, and I am showing only the mature villi. That's a tertiary villi. In some of the colleges they may ask you to show both primary, secondary and tertiary villi, but in the mature placenta we'll see only tertiary villi. The primary, secondary are the stages in the development of the villi. So I am showing the cytotrophoblast which are scattered, not continuous in the matured villi, because for the more exchange of gases, the cytotrophoblast cells will decrease in number. So the background, the smooth background, what I did was syncytiotrophoblast. I have to keep the blue nuclear there. Cytotrophoblastic cells, instead of continuous epithelium, they will be scattered. So here and there, cuboidal epithelium with borders, that's a cytotrophoblast. So you can draw the primary, secondary and tertiary villi representative of all the villi. The video is already been shared. You can see that video how all the three villi look. So I am keeping the round nucleus representing the syncytial and cytotrophoblast because they are cuboidal epithelium both. So this is the extraembryonic mesoderm, the dark shading, and which contains the fetal capillaries. The mesoderm may contain some connective tissue cells and half-bore cells, which are the macrophages, and fetal capillaries. So fetal capillaries will be lined by squamous epithelium. So please show the blue flattened nucleus lining the fetal capillaries. And inside the fetal capillaries, you can show RBCs. It is not compulsory to show RBCs. It's understood. I'm just keeping a pink dot in the center of the capillary showing the fetal RBCs. Adjacent to the villus is the intervillous space that will be filled with maternal blood. So this is the nuclei for the fetal capillaries. So the fleetoplacental barrier consists of this, that's the barrier between the maternal blood in the intervillous space and the fetal blood inside the fetal capillaries. So we can tell that you have a barrier from the fetal blood to maternal blood. It's a fetal endothelial cells, the basement membrane lining it, and the extraembryonic mesoderm, cytotrophoblast and syncytiotrophoblast. So this is the RBCs representing the maternal blood in the intervillous space. So this is the histology of placenta. You have to show the fetal side, the maternal side, in between the intervillous space. Better to show one stem villi. Now coming to the umbilical cord, and it is completely covered by amniotic membrane. It's a tubular structure. It is covered by amniotic membrane. So we just showing the circular structure, amniotic membrane. Amniotic membrane, as we know, it is lined by cuboidal epithelium or the flat squamous epithelium. So some book says it's a squamous epithelium. So write the cuboidal or squamous epithelium representing the amniotic layer. If you are writing cuboidal epithelium, please write the round nucleus in the center, squamous epithelium, flat nucleus. In the center, we have two umbilical arteries and one umbilical vein. Umbilical arteries, they have a crescent or star-shaped lumen. That is because of the elastic fibers and the condensation of the connective tissue in the subendothelial region. So So 2 arteries and 1 vein. The arteries have irregular lumen. That is mainly because of internal elastic lamina. They're similar to the arteries what we discussed earlier. So they'll have tunica intima and tunica media. One difference is there is no tunica adventitia. So they have an endothelium. You have to write the squamous epithelium, subendothelial connective tissue, and tunica media is full of smooth muscles. Some few elastic fibers may also be present, but majority is the medium sized artery. Umbilical arteries and medium sized artery, you have to show more of smooth muscles. So 2 arteries with star shaped lumen. You can remember as 2 eyes for the arteries and a smiley face for the vein. 2 arteries and 1 vein. It's like a face. Make sure to write the smooth muscles and occasional elastic fibers. Smooth muscle with a flat nucleus. Keep the nuclei for endothelial cells also. Coming to the vein. Vein is wider, like collapsed oval lumen. It also has a tunica intima and tunica media. Tunica intima has squamous epithelium. The media has more number of smooth muscle compared to the vein, which general vein. Here the umbilical vein carries oxygenated blood. Umbilical arteries carries deoxygenated blood. No tunica adventitia, just have to write the smooth muscles in the media, but thickness is compared to arteries, it is less thick. So surrounding the vessels, we have a mucoid connective tissue, gel-like tissue called Wharton's jelly, which has mesothelial cells, which are long stretched fibroblast. They'll be having processes, cellular processes. They're widely separated. You may also have allantois, part of remnant of allantois on one end of the umbilical cord. It may be present, may not be present. So these are the fibroblast cells. So flat nucleus you have to show, and connective tissue cells and gel-like structure. So in between the vessels you have fibroblast, fibers, collagen fibers, and also gel-like the background, the pink background. I hope the video was helpful. Please like the video, share with your friends, and subscribe to my channel. Thank you for watching.